Here we are. Hey, Mairead. I see your faces while we're warming up here. There's Akiela. Hey, Akiela, came. Elijah, that looks like COVID hair. Have you noticed, what's his name from The Daily Show? I forget. His hair keeps getting bigger and bigger. I think he hasn't had a haircut since the whole thing started. You're muted. You're muted. Elijah.
In all fairness, neither have I.
Nice. Nice. I wouldn't either, except it looks like hell when it gets longer, so.
I think you're talking about Trevor Noah.
Trevor Noah, that's right. Trevor Noah. Okay, what time is it? 1 o'clock? How many people are even in this class? I don't know. Hey, Nishi's here. Anyone want to give a shout out to Nishi? She said hello to us.
Hi, Nishi.
I'm just checking my roster here. Okay, that's enough Beethoven for now. So we got 35 people in the class, 29 have come. I think that's a record. Okay, so today's lecture is lecture 3. I just put the notes up about 10 minutes ago. I totally forgot to put them up earlier. So if you want to follow along with the notes, they are there. I am on an ether cable today, at least I think I am. Yesterday in cell biology, I kept cutting out. Hopefully that's not going to happen today. Okay, Aura, 101, 102. All right, well, why don't we go ahead and get started? I think this might be a little bit of a short lecture today. I'm going to talk about zebrafish development, which is in chapter 3. I don't talk about it again the rest of the class. I guess the main reason I put it out here is so you can just see the 4 different embryo models and their kind of macro changes, morphology changes during from fertilization up to neurulation. So with no more ado, I'm going to share my PowerPoint. Okay. Okay, there's the zebrafish. Note the lovely stripes on that zebrafish. There was a guy who was working in our department until I would say probably about 3 years ago, who was a developmental biologist, and he was trying to set up a system with zebrafish, but he didn't really work out, so he's gone now, which is unfortunate, but he really wasn't that good. Okay, zebrafish. Why do we like zebrafish? So zebrafish is a pretty popular model for development because it's got kind of a short life cycle for eukaryotes, for vertebrates. I mean, it's 12 weeks from fertilization to mating again. So in terms of vertebrates, that's pretty short. Like I think a mouse is about, I don't know, 6 months maybe. I don't know. I'm just making that up, but it's a good number. Also, it's easy to do genetic analysis with these things, easy to change genes in and out, transgenics and knocking genes in, knocking genes out. And they've got a transparent body, so it's easy to see internal structures during development. So there's a lot of reasons people like to use zebrafish. Personally, I liked Xenibus. I used to work with Xenibus years ago, and that was a really nice model. But here we are with Mr. Zebrafish. Maybe it's Miss Xenofish or Mrs. I don't know. Okay, let's take a quick look at the development of it. I've got some cool videos here, which are up in your Moodle page, but I think it'd be nice to watch them all together while we're here. Pausing. Okay. New share. Watch me be multimedia here. Okay, so here's Zebrafish development. So we're going to start out with at the 2-cell stage. Okay, so that's one big difference between this and Xenopus. Remember, Xenopus was a hollow ball of cells. This is like yolk with all the cells on top of it. So that tug into the interior is gastrulation. Somites. Look at those pretty somites. They'll be really important in this class. Okay, well, that's kind of nice. Okay, let me do another one. Part of it anyway. This guy does a really nice job.
Professor,
Yes,
you said somites become the spinal cord and the corresponding nerves?
correct. Well, not the nerves. No, hang on, stop. Not the spinal cord. They become the vertebrae. After neurulation, you get a neural tube, and that will be the spinal cord. And then the somites are there. Then there's there's a neural tube, and then underneath that is the nodal cord and the somites on either side of it. So the neural tube will be the spinal cord, and then the somites will become vertebrae that surround it, and the nodal cord becomes the squishy discs between the vertebrae.
Okay.
Does that make sense? Okay.
Yeah, it does.
Because keep in mind, nodal cord and somites are all mesoderm, so they're not going to be nerve tissue. They're going to be muscle bone. So that's why. Okay, can you guys see this new share here? Can you guys see Zebrafish development animation? Yes?
Yeah.
Okay, good. Let's watch this. This is about 5 minutes worth of worthwhile animation here. So again, just to contrast this to our Xenopus, remember the Xenopus egg, vroom, the whole thing split in half, and then vroom, the whole thing split in half again, so you got 4. Here, it's all happening on top of the yolk. And this is important because we'll see this in, I think, chick, but definitely mammalian, where you've got some internal cells and then you've got external cells. These are not touching the outside, these are touching the outside. And these will not be part of the final embryo. Only these will be part of the embryo. Okay, there's a new important vocabulary word, syncytium. So a syncytium is when you've got a whole bunch of cells, but then the cell membranes between them disappear. And now you've got one huge cell with many nuclei. That's a syncytium. Okay, so basically that lower layer of cells just kind of all fused together. Now you've got a whole bunch of cytoplasm, huge plasma membrane type cell, and a lot of nuclei.
Blastodisc is compressed down on the yolk cell. The yolk syncytial layer now consists of an external yolk syncytial layer, external to the blastodisc, and an internal yolk syncytial layer that is beneath the blastodisc. At the dome stage, the internal yolk syncytial layer begins to dome up, which is a sign that emboli has started.
So emboli is Australian for epibole. Damn Aussies.
Emboli is the process by which the yolk syncytial layer moves towards the vegetal pole and takes with it the blastoderm, so that the blastoderm spreads over the yolk cell. The development of the embryo is described by its percent emboli. The gastrula period commences at about 50% emboli. In the gastrula.
Okay, so remember, epibole is the ectodermal cells surrounding the whole embryo to give the skin layer. And then gastrulation is cells going inside to form the three axes.
Cell movement processes of involution, convergent, and extension dominate. At the shield stage, at about 6 hours, as involution continues at the entire margin of the blastoderm, the process of convergence commences. Deep layer cells, in addition to moving towards the blastoderm margin and involute, they also converge towards a single position of the germ ring to accumulate to form the embryonic shield. At the embryonic shield, the involuting cells form the axial hypoblast that continues the movement of involution to stream towards the animal pole. At 90% emboli, the axial blastoderm thickens on the dorsal side rather than the ventral. At 100% emboli, bud stage, at about 10 hours, the gastrula period is defined as ended. The emboli is complete. The tail bone has formed.
Okay,
Embryo development
I'll stop
begins
you.
with fertilization.
We've had enough of you. Okay, so that was kind of neat, I thought. Shut up. Shut up. Okay, so that's kind of what we're going to talk about today. I'll go a little bit slower, and we'll see it in a different way, but the same thing basically. What's going on in chat? Is there supposed to be a sound? There was no sound. Anybody hear sound?
There was like sound halfway through the video, but for the first half, you couldn't hear anything.
Okay, well, there's a link to that video in Moodle, so you can check that out again. I say it's the one that says start at 250, because the first half of it is about fertilization and stuff, and we're not even talking about that. So check that out if you want to see it. I think you should. It's nice. Okay. Bum, ba, bum, bum, ba, bum, ba, bum. Okay, doke. So, okay, so the one big difference here between this and Xenopus is after fertilization, basically all the cytoplasm goes to the top and all the yolk stays on the bottom. So you get really one cell on top of the embryo or zygote, zygote. And then that splits to give you 2 cells, and that becomes 4, becomes 8, and these are all on top of the embryo. Where are the lecture videos being posted? So they will be posted in Moodle. Did I post the last one? I'm not sure I did. I'll make sure they're up today. I don't know if there's anybody here from cell biology, but I didn't get a recording from cell yesterday, but this I will get up there. Okay. Okay, so again, different from your Xenopus, you go all the way to the 64 cell stage, and all the cells are in one on top. There's 2 layers of cells here. Now the next thing that's going, so let's look inside at those cells. Okay, so it's like a hemispheric ball of cells. Once they divide and you get a whole bunch of them, you've got a hemispheric thing. Now you've got the enveloping layer, which was pretty distinct in that video. Here it's a lot thinner. So those cells, again, will not become part of the embryo in the end. They started as part of the embryo. Now they're going to be external structures. And then you've got ectoderm on top, mesoderm next to that. And these next type of cells are really kind of different. They're mesendoderm. So the third type of cells here is not endoderm, but mesendoderm, because they can go either way. Each cell can go to endoderm or mesoderm, and that will be specified after it goes through gastrulation. There's an important word too, specified, specification. Keep an eye on that word because I use it a lot. It means changing cell type and making it more specific. So remember, these 3 cell types are very general. They can become a lot of different things. You can call them pluripotent because they can become many different things. Specification makes them less potent and more specialized. Okay. So let's see, inner cells are the deep layer, and they're the ones that form the embryo. So the ectoderm, you've got ectodermal cells, mesodermal cells, and mesendodermal cells. And then underneath that, you've got this yolk syncytial layer, which again is kind of like there's a cell membrane on top and on bottom, and a whole bunch of cytoplasm and a bunch of nuclei. It's just one big mass of goo. Let's call it goo. I think that's a technical term. And then epibole starts. And epibole is again the ectodermal cells growing down in 3 dimensions around the outside of the embryo. Now, as they go down and down and down, they'll reach a certain point where they start to involute and build up. So now they're building up here, like again, in 3 dimensions all the way around. You're getting a thickening here. So you get this thickened layer, and especially over here, they're thicker than over there. This ends up being the dorsal side of the embryo. And another theme we'll see throughout the course is when you get right before gastrulation, you'll get one area that will become dorsal, and that will then specify other cells to become what they're going to be. So this is a really important bunch of tissues we'll call the organizer. The organizer are the cells that will change other cells into something else and give you those three axes that we're talking about. They're going to cause the gastrulation and everything. Okay, so mesoderm and mesendodermal cells will move down, and once they hit this germ ring, they're going to involute and go into the, and go into the embryo. Now, again, one slight difference here between here and the xenopus. In xenopus, all of the involution happened at the dorsal lip. Everything came in that way and went across, which I think is a beautiful thing because it makes it easy to follow, easy to understand. Here, in 3 dimensions, all the way around this ball, cells are going down and in. But it's preferentially happening over here at the dorsal side. So basically what's happening is cells are coming down the side, involuting, and then crawling across the ectoderm. Okay, you saw that last time with the xenopus, but now it's a little more 3-dimensional. Okay, occurs all along the periphery, starting at the shield region. So the shield region is this edge here that goes all the way around the thing. Mesendodermal cells at the lateral edges now, those mesendodermal cells will become either endoderm or endoderm or mesoderm. And so that's where they're getting specified to be one or the other. And then they'll spread up. Ectodermal cells are still dividing, and they're producing epiboly, covering the whole embryo with skin. And now we get this process called convergence and extension, which is happening here. Let's see what our next slide looks like. So this is one way of looking at convergence and extension. Again, throughout this class, visualization is hard because you're looking at a 2-dimensional figure and trying to understand it in 3 dimensions. But basically, all these endodermal cells are coming around and into that shield layer and then coming together and moving forward. You can't even see me. Well, coming forward like this. So they've ripped off the ectodermal layer here, so you can see these mesodermal cells are coming towards the center line, coming together, and then pushing forward like that. That's convergence and extension. A little easier to see here, I think. So again, this is that organizer here. The cells are coming together into the center, and once they come into the center, they push forward across the top of the embryo underneath that ectoderm, and they're forming your 3 axes, okay? This will be the dorsal, and then anterior. It's always going towards the anterior, posterior back here, and then right and left. Here's another view of it here. There's your shield. Cells are coming down and in, coming down and in and together. Here they're converging, and here they're extending forward. Okay, convergence and extension, that's gastrulation in the xenopus. Okay, so gastrulation in the, I'm sorry, not xenopus, zebrafish. Let's go back just for a second and look at gastrulation here. So what's different here with your xenopus is it's all happening here. So you don't have cells all over the whole ball coming in. You only have cells that are moving towards this blastopore lip and moving in. So that's the big difference between our xenopus and our zebrafish. Okay. How's that? Everybody happy with that? Anyone want to send me a chat and tell me where it's confusing? We're all good? Okay, I was saying this is going to be a short lecture, and I think it is. All right, so the next step after gastrulation, what comes after gastrulation, Lee Turner? Oh.
Neurulation.
Neurulation, excellent. After it gastrulates and you end up with this with the intestinal tract, the next thing is neurulation. Okay. Gastrulation. Okay, so again, comparing Xenopus and zebrafish. In the Xenopus, we had the neural folds come up and the neural folds met and fused together, and you ended up with a neural tube and neural crest cells. Here, it's a little different. It's still happening over the notochord, and you've got neuroectoderm on top and epidermal ectoderm on the sides, and we're folding up again, but instead of leaving a hole in the middle as it folds in a closed way. So as it folds, it keeps closed. So they call this the neural keel because it kind of looks like the keel of a boat. If you know anything about boat building, the keel is put down first, just a big long wood thing in the middle, and everything's built off that keel. So they call this the neural keel. And this continues to close until it totally closes up. And now you've got the neural rod, okay? Instead of neural tube, a neural rod. And the epidermis keeps moving forward until it encloses the whole back end of this neural rod, or the dorsal side of the embryo. And then eventually you do get the hollow part. So it turns into a neural tube, but this is afterward. That part in the middle gets filled with fluid and becomes a neural seal. Okay, seal is always your cavity in the middle of something. Okay, that's it for zebrafish. That's about all I will ever say about zebrafish. I think what's most important here, what you might see on an exam would be what's the difference between Xenopus and zebrafish in terms of their gastrulation or in terms of their neurulation. That's kind of how I would put the question, because if you can answer that question, that means you know both Xenopus and zebrafish for both of those processes. So 4 processes for 1 question. That's a lot of bang for my buck. Okay. To review that a little bit, here's your Xenopus, blastula, hollow ball of cells with a marginal zone that has the endoderm, and the bottom is marginal zone is mesoderm, bottom is endoderm, and they're separate. The endoderm and mesoderm are separate individual cells. You start to form this dorsal lip, and then you gastrulate through that dorsal lip. So the cells here are coming in and through and over the top. And that proceeds in the anterior direction until you've got the archenteron, which is the primitive gut. The blastocele is going to be forced out through the yolk plug over there. And that's your Xenopus gastrulation. In zebrafish, you start out with a, this is called the blastoderm, or I'm sorry, I should know this. Blastoderm, yeah. Why do I ever doubt myself? I'm so rarely wrong. So you've got a blastoderm instead of a blastula, because it's all on top. It's not, you know, in a ball shape. And you've got your ectoderm, mesoderm, and your mesendoderm. And then that epibole starts and the ectoderm pushes down, and then the shield region forms. Cells converge and extend through that shield region on all 3 dimensions, but mostly on this side. And then it converges on this side and extends through to the anterior portion, and the head will end up over here. Okay. Questions? Preguntas? Nunca. Oops, I'm going the wrong way. Okay, no questions. We're going to move on. I've still got 20 minutes. I don't think I'm going to take the next 20 minutes. I'll take another 10 and set up our Friday's lecture. So Friday, we're going to start in on avian and mammalian development. And by avian and mammalian, I mean chicken, mouse. That's all we're talking about here. So, okay, how are these going to be different from the previous ones that we saw, the Xenopus and zebrafish? For one thing, you're not going to have a a ball. Okay, you're not going to have a ball like we do here. Remember well, especially with the mammalian comparison, your xenopus and zebrafish are both externally fertilized, and then you've got an egg that develops all on its own. What does that tell you? You need a lot of yolk. So yolk is a big important part of those embryos. Here, chick also has a lot of yolk, but it's getting more towards a placental type model, sort of. And then the mammalian thing doesn't have much yolk at all because pretty soon it's going to have a placenta and it won't need it. So you don't start out with a big ball of cells wrapped around a yolk. What you end up with is like a flat disc of cells, multiple layers, but a flat disc. And we call it an epiblast. So this is our epiblast. And then we'll talk about the zona pellucida and zona opaca later. They're important because you've got different cells here and there. We'll get into that. But let's see. What else? Cells continue to divide during gastrulation. Okay. With these other ones, the cells kind of, especially xenopus, the cell division stops for the mesoderm and endoderm, and the cells just move. And once they reach their final position, then they start dividing again. Here, they'll continue to divide as they're gastrulating. And so as they're gastrulating, you've got a mix of cells, endoderm and mesoderm. And gastrulation will occur along a primitive streak. So when you're looking at this in the microscope, you can see the streak extend from the posterior to the anterior direction. Posterior to anterior? Yes, posterior to anterior direction. And then cells, that primitive streak involves cells involuting through it. So they're coming through and out like that. So the cells enter the epiblast and converge on the primitive streak. The center, okay. So they're coming from the outside and they're coming down this primitive streak, and they're doing this as individual cells. So we don't have, like in xenopus, a whole sheet of cells pulling along. Here we've got cells coming through and then they escape from each other and they migrate individually, okay? And at that point, then the bottom layer becomes endoderm and the middle layer becomes mesoderm. So they change as they go through. And the specification occurs in this region here. Again, this is kind of like the organizer. This is going to give off signals that will cause specification to happen. So the cells are dividing out here, and then they're pushing through this way, and they're specifying into mesoderm and endoderm. They're becoming individual cells crawling. We call that mesenchymal cells. I'll talk about them later. And then they crawl off and go into position. And then the gut, if you remember from xenopus, the gut was formed by gastrulation, right? When the cells gastrulated, they left that archenteron there, which was just a gap between two layers of endoderm. That doesn't happen here because you can see these cells just kind of individually go through. So after the endoderm layer forms, it then opens up into a gut. So the gut is made differently. Okay, so that's going to be the big thing here with chick and mammalian, the chicken mammalian model, the primitive streak. Another thing that's going to be really weird about the mouse, really weird about the mouse. I don't know how mammals ever evolved because this is so weird. So you can see this primitive, epiblast is like this flat thing. You can imagine, oh, that could turn into something. Well, in the mouse, it like flips inside out and forms a cup. So it's a cup-shaped embryo before gastrulation and during gastrulation. And then for neurulation, it has to flip inside out. Really weird stuff. So I'm really looking forward to explaining that to you because I don't know if I understand it. Just kidding. So we still have 15 minutes left. Let me see. Let's stop the share. What do I have for you? Let me see if I can do a little, if I can annoy you somehow real quickly. I hadn't planned on doing this, but I'm just looking at a quiz to see if I can throw something at you. No, I'm not going to do that because it's too on the fly, and I'm going to give up important questions that I won't be able to ask you again later. I'm being selfish, I admit it. Okay, any questions, comments? Again, a little bit short today. The hard thing for me about Zoom, if you had me last semester, is it's always like if I'm in class and talking to you person to person, I'm writing notes on the board, and that takes time, and it kind of slows me down, and we get to kind of live with the material a little bit longer. Here, it's like boom, 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 PowerPoint, 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 and then we're done. So it tends to be a little bit shorter. And so I really suggest if you don't understand something as we're going through, ask questions. Just throw them up on the chat and I'll answer them. And that'll slow us down some, and hopefully we'll catch more people as we're going through so people won't get lost. Okay, any questions, comments? Okay, this is it. I'm about to say goodbye. All right, I'm shutting her down. Have a great afternoon. Go get something to eat. See you later.
Thank you. Have a good one.